Well, it's interesting. There's been a lot of excitement over the last 5 or 6 years about immune checkpoint blockade in multiple cancers, including kidney cancer. And nivolumab has been approved in the US for several years for patients who failed VEGF therapy. So there was a lot of interest of trying to bring immune checkpoint blockade from second line into first line. And there have been a number of different strategies to try to get that accomplished by essentially combining first line and second line with VEGF and PD-1. There's a lot of interesting data at this meeting about those approaches. And also combining PD-1 with CTLA-4 blockade with IPI and Nivo, which is obviously now approved in the US and was recently, granted some step towards approval in Europe. But little was known about single-agent PD-1 blockade, so we said, let's try to figure out how active is PD-1 in untreated patients, how much of this benefit that we're seeing with combinations is driven by PD-1. And it turns out that it's a fair bit, So last year at this meeting, we presented cohort A of the KEYNOTE-427 study, which looked at clear cell patients. So about 105 patients or so were enrolled on that study with single agent pembrolizumab in untreated patients, and the response rate there was 36%, which was actually higher than we expected, higher in intermediate and poor risk patients, and in PD-L1 high patients, the response rate was actually 50%, suggesting maybe you might be even able to select for those patients. So that was impressive, and it helps put some context around what we're seeing with combinations. This year, we're taking it a step further, looking at patients with non-clear cell kidney cancer. So these are patients who truly don't have many therapies. And in fact, when it comes to immune therapy, they've been excluded from most of the trials we've seen so far, in part because sort of historically they tended to respond less well to cytokines. So people assumed they'd be less likely to respond to immune checkpoints. And it turns out that there's some activity there. So when we looked at single agent pembrolizumab in cohort B of this study, same design, same dosage, just different types of patients, we actually looked for non-clear cell and then we confirmed it with an independent review. Most of the patients on the trial were papillary, but we had chromophobe patients and unclassified patients. When you take everyone together, the response rate was 25%. 25% in papillary, lower in chromophobe, 10%, higher in unclassified, 34%, overall 25%. We actually saw a CR rate of 5%, which was interesting. We are seeing complete responses in non-clear cell kidney cancer, and we took it a step further, looking at, once again, PD-L1 in the microenvironment by immunohistochemistry. And there, if you were PD-L1 low, less likely to respond, 10% response rate. But PD-L1 high, the response rate was 30%, so 3 times higher. So in some ways, the IHC test actually performed better in non-clear cell than in our clear cell patients, suggesting even here that it's probably more the biology, the reaction to the tumor that drives the benefit of these checkpoints, rather than the actual tumor histology. So, interesting, we saw, survival, median survival hasn't been met in this cohort. Reasonable tolerability, as seen in many pembrolizumab trials. The percent of patients who came off drug for toxicity was relatively low, only 6%. However, there were 2 treatment-related deaths, so we obviously have to monitor our patients very closely. But it suggests that for these patients who have very few options, that single-agent PD-1, in this case with pembrolizumab, might be an effective option for them. And we're going to, try to make it more available to them in the future. Well, it's a very interesting question. There's not a lot of head-to-head data, so most of what you say is sort of speculative. But essentially, the data in the first-line single-agent experience with pembrolizumab was better than we expected. We need to get more single-agent experience with these other agents, and obviously, head-to-head trials would be nice, but we're stuck making sort of cross-trial comparisons, which are fraught with all sorts of potential biases. But in general, the data with single-agent PD-1s is hard to beat, when you do a cross-trial comparison. It doesn't seem like, at least in kidney cancer, that PD-L1 strategies produce as much efficacy as PD-1, although that's, sort of a speculative stance going forward. And that's going to be a controversial issue going forward, is like, which are the preferred combos that are based on PD-1 or PD-L1. There'll be a lot of debate around that question going forward.